Yeah, I think we might make a start with just some introductions and brief presentation on OBON, just to get people up to speed with what OBON is doing as a program. So welcome. I'm Sophie Seave. I'm the CEO of POGO, the Partnership for Observation of the Global Ocean, which is the lead organization for OBON. And my colleague Luz, who is the OBON program manager, will be giving the introduction to OBON, just to, as I said, get people who may be new to the program up to speed with what OBON is all about. So it will go into the presentation, and then I'll introduce the speakers afterwards, if that's okay. Luz?
Thank you, Sophie. Hello, everyone. As Sophie mentioned, I'm Luz Rodriguez, and I'm the OBON program manager. So, can you see my presentation now?
Yes, that's perfect.
Okay. Okay, again, welcome everyone to the Ocean Biomolecular Observing Network. Again, a program endorsed by the UN Decade of Ocean Science and led by the Partnership for Observation of the Global Ocean. The next one. Our strategy is aiming to work towards global biomolecular observatory supporting a sustainable ocean. And we would like to invite you to visit our website or contact us to learn more about the program. So you can find us in this URL, and or just scanning the QR code that you see on the screen. In a nutshell, OBON's mission is to accelerate informed decision-making to restore the health of our ocean using the universal signatures of life on Earth, biomolecules. And our mission is to create a hub for biomolecular measurements of marine life. We are guided by 4 objectives. First, to build a coastal to open ocean biodiversity observing system using multi-omics and autonomous tools. Second, to grow capacity through training programs coupled with funded equipment supported by aid agencies and philanthropy. Third, to strengthen marine omics data systems and improve ecosystem models by adding biomolecular insights. And fourth, to partner with conservation experts, policymakers, and the private sector, so this work directly supports sustainable development. This diagram here illustrates OBON strategy, which was launched earlier last year, and it shows how it is articulated around these areas, which are aligned with the 4 objectives I just presented. It aligns by developing networks and resources, developing and innovating the technology required and enhancing the use of information from biomolecular observations and ultimately informing policy for a sustainable future. As a UN-endorsed program, we have hosted 25 projects. 23 of them are active at the moment, and we are waiting to involve another few from the last call. We are gathering information on the sampling location that all these projects cover, achieving global coverage. We do have mostly coastal sampling. Oh, sorry, it's my keyboard. Also, we do have mostly coastal sampling locations. In each of the UN Decade calls, we are encouraged we are encouraging the endorsement of open ocean projects, and we are open to collaborate with one-off research cruises that might not be sampling regularly, but that now can be endorsed as contributions instead of UN-decade projects. How are we organized? The Program Coordination Office is hosted by POGO, and the program receives guidance from a scientific advisory committee and an executive committee, both chaired by Margaret Leinen. We are in the process of setting up a user advisory committee coming this year to ensure participation from different sectors towards our goals of accelerating the use of biomolecular science and technology. The OBON projects work together through 6 different working groups, which involve activities and discussions around these topics that you see on the screen. They meet online a couple of times a year, and most times they gather in person at the OBON annual meeting. Before I finish, I would like to share a few of the latest activities of the network, which are the workshop Leveraging Biomolecular Technologies for Supporting Sustainable Fisheries and Aquaculture, which we run at the beginning of September at the FAO headquarters in Rome. The Latin America eDNA Network and Capacity Development Workshop, which is happening in Colombia, and the second Marine Omics Technology and Innovation MOTI workshop, which will take place in Hobart in November this year, within the framework of the OBON annual meeting. Here you can find our list of contacts. You can follow us on our social media or reach out to us directly. We would be very happy to hear from you and your projects on marine biomolecular science. Now I will hand it over to our first presenter and to Sophie first, I guess. Thank you very much.
Great, thank you, Lewis. So indeed, we will now move into the presentations, providing updates on OBON endorsed projects. And just for context, we're recording this series of webinars and making the recordings available on our YouTube channel, both to introduce OBON and its projects to the broader community, but also to allow the participants in the OBON annual meeting to kind of get up to speed on the latest project updates ahead of our meeting, which will take place in Hobart in a couple of weeks. So the first presentation will be Kristen Westfall from the Department of Fisheries and Oceans, Canada, who will be presenting an update on the project Marine Biodiversity Library and MPA eDNA monitoring, which has been an OBON endorsed project for, I guess, about a year and a half or so.
Yeah,
So,
that's right, Sophie.
Kristen, if
Thank
you'd like
you.
to share your screen.
Thank you, Sophie. And how is that looking?
That's looking great. Perfect.
Great. Okay, so thank you for the to OBON and to POGO for the opportunity to speak today. Let me go back to my title slide here. It's not wanting to work. Okay. Okay, I'm just going to get out of this and I'm going to share from the beginning. Sorry about that. Here we go. Let me just try this one more time. Okay, there we go. And that works perfectly. Wonderful. Okay, so building, sorry. Building eDNA metabarcoding tools for biodiversity monitoring in Canada's MPA. So this is sort of how our title has evolved over the last year and a half, and it includes the reference library that we've been building. So this is a project with myself and my colleague Emily Rubidge at Fisheries and Oceans Canada. We're at different institutions on Vancouver Island. I'm in Nanaimo and Emily's in Victoria. So the main project objectives are focusing around marine conservation genomics and biomonitoring. Our first main objective is developing eDNA metabarcoding tools for ecological monitoring and integrating those tools and the data that they produce into MPA management and monitoring frameworks. And finally, bridging science, policy, and technology. So turning eDNA data into actionable conservation data for the Government of Canada and other groups that co-manage MPAs within Canada. We have a few MPAs that are co-managed amongst different groups. And so why do we choose eDNA as an ideal tool for monitoring marine protected areas? Well, Canada is a large country with a large EEZ, large marine territory, and we have signed on to protect 30% of by 2030, and this will include a large amount of our marine territory as well. And so far, we have over 800,000 square kilometers of marine protected areas and OECNs, which is anything that's protected not under the Oceans Act. So this is other effective area-based conservation measures. And most of them are remote, they're large, and they're deep, and which means that they are data deficient for the majority of them. And eDNA offers a non-invasive, a scalable, and a multi-taxonomic efficient tool for detecting biodiversity and monitoring biodiversity over time. And so that's why we're looking at eDNA as one of the main tools that we're going to be using for monitoring marine protected areas over time. So this isn't necessarily monitoring the effects of the conservation, but looking for changes in species distributions, effects of stressors, effects of climate change, and whatnot. So what we're doing now, the phase that we've gotten to in our program is we've been spending a few years doing a snapshot biodiversity assessment along the Pacific coast of Canada. And now we're transforming that into a long-term monitoring tool. And that's going to be the majority of my talk today. So the Pacific eDNA biomonitoring program has been running since it's part of our larger OBON project. It's been running since 2022. The main objective of this program is to build a national framework for biomolecular MPA monitoring. So although we do most of our work in the Pacific region, we have partners across Canada working in MPAs in other regions, and we work together to build eDNA as a tool, useful tool. So from 2022 to 2024, most of our time was actually spent really just going out and collecting a lot of data, a lot of metabarcoding data. We called it our rapid biodiversity baseline phase. And we collected over 1,800 samples on the Pacific coast of Canada. The map on the right here is just showing where all of the samples were taken. The purple outline are marine protected areas. And then in the green, we have proposed MPAs. I'll get into these specific MPAs a little bit later. And the dots are colored by survey year. And so these first 3 years were really just spent nailing down our laboratory and bioinformatic protocols for large-scale operations. We've also developed an eDNA sample biobank from multiple habitat types across the British Columbian coastline. So most of my talk's now going to focus on phase 2, which is what we've been doing in 2025 and how we envision the project going forward. And so this is basically taking the data that we've been collecting for the last 3 years and turning it into a tool that we can actually use for to incorporate on long-term monitoring frameworks. So in order to do this, we're focusing on specific conservation priorities for marine protected areas in Pacific Canada, and we're demonstrating the tool performance across multiple ecosystems and different taxonomic levels. So I'm going to talk about phase 2 in 2 specific use cases for marine protected areas on my coast. The Great Bear Sea MPA it's not yet enacted. It's a very unique marine protected area in Canada in that it's been co-developed by the Government of Canada, the Province of British Columbia, and 17 member First Nations groups. And so it's taken quite a while to co-develop the network. On the right-hand side is a map of the footprint of the of the network. It's also known as the Northern Shelf Bioregion. And last year, they published a network action plan that identified over 140 single species conservation priorities for this MPA. And it's expected that this MPA will be enacted in the next few years. And so in order to determine if the tool that we've developed over the last few years will be efficient at ongoing monitoring for this MPA, we looked at how many species of these 140 conservation priority species could be detected with our tool. This was a preliminary assessment, really only based on data from 2022, and we're analyzing data from 2023 and 2024, and that will be in our larger report. But from data just from 2022, we discovered that 74% of these 140 species can actually be currently detected, or they're missing from the reference library. So they're an easy add. So we can go out and specifically sequence the species that we're missing, and we'll be able to detect them because there wasn't any primer mismatch or whatnot. So theoretically, if the species is in the region, we would be able to detect it with the markers that we're using. The graph on the bottom is, sorry, I should have said this earlier. This is the distribution of these 140 species across 13 major groups. I've broken fish down into bony fishes without rockfish and rockfish, just because rockfish are a special group that I'll talk about it right now, actually. So the major gaps where we are not identifying as many, a large number of species in that taxonomic group is rockfish, marine mammals, crustaceans, and mollusks. And we knew this going in. Rockfish are a challenging group to detect with eDNA because they're genetically very similar. And so the regions that we're using, the 12S and 16S regions, don't discriminate among most of the species. And we have about 45 species of rockfish on our coast. And so we've actually applied a specific rockfish marker developed in California in the cytochrome B region called Mycevastes. And we have applied this in our 2024 data, so that those results are still to come. And marine mammals, crustaceans, and mollusks, I'll get into in the next slide. So we're identifying the gaps in order to be able to operationalize this tool for monitoring. So this graph goes into more detail about the species that weren't detected and the reasons why. So the height of each bar is the total number of non-detections for that taxonomic group. And the different fills are the reasons why. So for rockfish, they're not resolved to species. And then we have no reference DNA sequence, mainly for crustaceans and mollusks here. And then we also have where primer mismatch would dictate theoretically that even if the DNA was in the water, that it wouldn't amplify. And so we can see that there's easy access to these crustaceans and mollusks here, where we could add those species to the reference library. and potentially detect those species. And then, as I said before, we could apply specialized markers to this rockfish group in order to resolve down to species level. But for the species where these reasons, these potentially false negative reasons don't apply, we've put those species out on in this table here. And the majority are marine mammals, and the majority of they're likely to be true negatives in most cases, because the species just don't exist in the areas that we were sampling. So some of these marine mammals are extremely endangered or extremely rare in British Columbia waters, and or they're present in offshore habitats which weren't sampled in 2022. Okay, so I'm going to now jump into the next use case. So this is focusing on deep sea environments off the British Columbia coastline. This is a collaborative effort between myself and Emily, who are co-leads on this project, and the Deep Sea Ecology Program, also at Fisheries and Oceans Canada. And they run the Northeast Pacific Deep Sea Expedition. And so the large marine protected area on the map on the right-hand side here, this is the Tungwan-Hutchcock-Segas MPA. And this was a new MPA enacted last year. And it's Canada's largest marine protected area, covers 2.3% of our national marine territory. It is an extremely important marine protected area, not that they aren't all, but all of the known Canadian hydrothermal vents are in this NPA. It also includes 47 seamounts and other ecologically and biologically significant areas like cold seeps and deep sea ridges. And the second MPA, which is quite a bit smaller up on the top here, this is the Ascan-Kingless-Bowie seamount. So this is a single seamount, sort of a collection of seamounts. And together, these are our offshore Pacific bioregion use cases. And so for the deep sea, because these are even less, more data deficient than coastal systems in British Columbia, we have spent a lot of time developing the reference sequence database. And we've been doing this in conjunction with NEPDEP, the Northeast Pacific Deep Sea Expedition. So on the bottom of the screen here, this picture is of the picture on the right is a special remotely operated vehicle called ROPOS. And it has, I don't know the depth rating, but we've had it down to 3,000 meters. And it has water collection devices for collecting water for eDNA. And it also has robotic arms to collect specimens. And so what we've been doing in these deep sea MPAs is doing transects for taking video, collecting water in when we come across really interesting habitats, or like we come across a hydrothermal vent, collect water, and then also collect specimens for species that either are new to science or are just very understudied. Or and those specimens have been sequenced in our reference library. So now we're pairing our eDNA samples with newly generated reference sequences in order to improve the detectability for eDNA as a tool to monitor these environments. And so it's really important to develop the reference database in an environment like the deep sea, because it's just so data poor. And so that brings me to talk more about this reference database. So the Canadian Marine DNA Library is part of the larger OBON project. And we developed this specifically because other genetic repositories don't quite meet the needs for eDNA metabarcoding that may have specific management objectives or regulatory objectives. And so for CAMDL, we focus on genome skimming to generate full mitochondrial genomes for all the species. We also have standardized metadata that adhere to FAIR data standards. And all of the sequences are tied to voucher specimens. So this is one important aspect that is definitely not present in a lot of existing the large global genetic repositories. So all of the specimens are cataloged in Canadian museums. And then we also built a scalable web platform that has search and download capability. So this was a pilot project where we had just 2 years of funding, the majority of which went to the website. So, and we also were able to collect and sequence close to 200 specimens, and this is the taxonomic distribution of the specimens that they're almost ready to be fully uploaded to the database. So on the bottom here, we have almost 60 species of corals and sponges. The majority of these are deep sea corals and sponges from those ROPOS expeditions. So like I said before, we've got standardized metadata, and these are in line with international standards. So 55 metadata terms are associated with every specimen in CAMDL, and the majority of those are from our Darwin Core terms. And then we also have specific genetic terms that come from Mixis. And this is a screenshot of the homepage for CAMDL. The heart of CAMDL is this database. So here, if you go to the website, this is the URL here, you can search the CAMDL database. It's not fully populated yet, so I would recommend it to search it in a couple of months. And you can search by taxonomic rank, you can search by region, and in the search and you can search by gene of interest. So you're not downloading full metagenomes because normally in eDNA we don't sequence full metagenomes. So if you want your CO1 gene or your 12S gene, you can just download the genes that you want or the species that you want. And in the search results, you can pick and choose which species you want to download. And then you can download a FASTA formatted file, and you can also download the metadata associated with those sequences. Okay, so that leads to where is this all going? And like I said, we've spent, excuse me, the majority of the last few years we've spent on the ground collecting data. We've spent it improving our laboratory and bioinformatic workflows, but there's also a lot of other work that goes on in the background in order to make eDNA an operational tool. And that's surrounding things like standardizing all of our sampling and laboratory and data workflows. So not only are we optimizing, we now have to think about standardizing, especially if we want to implement eDNA as a nationwide tool for in the management realm within DFO. And then we also have to work with the data in order to determine if eDNA is a good tool for assessing conservation performance indicators. And of course, there's always the data issue when it comes to genomic data, and strengthening the genomic infrastructure for Canadian biodiversity and also on the international platform. And OBON is a really great group for strengthening these international or making these issues also working together on an international scale. And of course, fostering partnerships. So in Canada, we work alongside First Nations and NGOs in order to co-develop and co-manage our conservation areas. and international networks like OBON and OBIS as well for data management. And with that, I'd like to put up the many acknowledgements for this project. And I don't know if we have time for questions, Sophie. I'm certainly open to that if there's time.
Yes, thanks very much, Kristen. I think we have a couple of minutes for questions, especially since you will need to leave before the end of the webinar. So, are there any questions for Kristen?
I have one, if I may. I'll move to
Yes, Jay?
We've been working with eModNet to, in the metadata for data, the DOI or the description of the practices or methods used to collect the data. And you guys are doing remarkable stuff. I mean, OBON has a best practices activity and has for some years. Are you looking at documenting the methods? Are you looking at putting in the metadata the description of the method used to collect that data?
Yeah, absolutely. So we've adopted the FAIR E standards put out by Miwa Takahashi, and so that will include links to all of the protocols used and all of the specifics for the assays and the bioinformatics as well. So everything we are fully supportive of reproducibility and transparency, and so absolutely we will be publishing all the metadata surrounding not only the samples but the project as well.
Oh, that's great. We do have a repository for methods and best practices in the IOC, the Ocean Best Practice System. And
Right.
if you would like to have a copy, it doesn't need to be uniquely there, but have a copy of the methods there, we would encourage it and help you with that.
Oh, that's great. Thank you.
Okay, thanks, Jay. I'm not seeing any more questions, so I think we'll move on to our next presentation. And thanks again, Kristen. It was great to hear all the progress made since this time last year.
Yeah, thanks.
So, okay. So our next speaker is Jennifer Sunday. So we're staying in Canada. Jen is from McGill University and will be providing an update on the Pacific eDNA Coastal Observatory, which is one of the fairly early projects, I think, in OBON. It's been endorsed for about 3 years now. So we're seeing your screen. There we go.
How's that look? All right, great.
Yeah, perfect.
Thank you so much. I don't know, I'm just going to dig right in. And thanks, Kristen, that tees us up really nicely. We are going to extend a little into more of the Pacific. So this is the eDNA Coastal Observatory. And yes, it's been a member of OBON for almost since the first, maybe the second or third group. So I'm just going to run through a little update and then hopefully there's time for questions. So we call ourselves PICO, it's the Pacific eDNA Coastal Observatory. We are focused on this area of the ocean, which is the Eastern Pacific, really near shore. So this was a network that established really quickly during COVID. And every group is going out into seagrass habitats. You see some pictures here. Some of these are researchers who have done long-term monitoring for fish or for invertebrates in those seagrass. Others just jumped on to help us balance out our spatial balance across this transect. And we especially tried to make sure we were sampling, or we had nodes above and below major biogeographic breaks, so that we could do both biogeography and then also start to track species range shifts over time. And in every group, every regional node samples, the design is that they all go to 5 seagrass habitats, and within each they sample 4 water samples and a negative control. So it's both a fair amount of I don't know, it's either a lot or a little depending on your perspective, but it is balanced, and you'll see we try to accumulate the number of species that we're looking at. So far we're only looking at fish. We started in 2021, so we've just finished the summer of 2025. We go out in 1 month every summer. And we have, in the first year, we also did beach seining at just over half of the sites. So we can compare. So this is the kind of habitat where you can drag a net and catch the fish. So we can relate the fish that we're seeing from beach seining to what we're seeing in those 20 eDNA samples. And you can see some regions blink on and off, where it's very voluntary at this juncture, although we try to also work on how we're sharing capacity and how we provide data back with all of our partners. Here's some pictures of the operations. If anyone's interested in this kit, it's very low cost, ships light. If any parts of it are missing, people can get them locally, the parts. But it still does a positive pressure through a Sterivex filter for the nerds out there. So we try to also maintain, so we do that because we want it to be really low chance of contamination. So no details with fiddling with filters. Happy to talk about that more. And it all ships in these totes. So they go out to the partners and they come back with the samples in buffer, filtered and stored in buffer. So we have videos, and we talk to our partners. Here's a big list of the leads from every region. So it's a lot of communications, but it's super fun, and it's starting to be really exciting what we're finding. So I'm gonna get to that. Oh, here's our pipeline. So just to say to the note about protocols, if you see this little symbol, these protocols are published and available. We've gotten all the way to just our bioinformatics pipeline, which we're about to publish, and then the whole thing will be shareable and reproducible and findable. Don't know. Okay, yes, and very important to mention the Hakai Institute. So the Hakai Institute is where the kits are prepared, bleach cleaned, and then sent out to every partner, and also where everything's returned to. So we do all of our lab work in-house at the Hakai Institute. So it's all done in one place. For better or for worse, for now, that way all of our data are immediately comparable and interoperable. Although it would be really interesting to send some samples around to other labs to do some comparisons. Okay, so here's the kinds of things that we're finding. So this is from 2022. I'm going to show you all the years, but just to give you a sense that this is about 147 fish species on the x-axis, and on the y-axis is the list of regional nodes. And so you can see already there's this biogeographical pattern where you have southerly fish species that are southern affinity, moving into a more northern affinity fish fauna. Here's 2022, and I'm going to show you like the full set, 2021 to 2024. So this is now feeling like a data cube. We can look at species ranges as we look up and down this way. We can look at species assemblages as we look horizontally, and then we can stab them through time and see how any of those species ranges or assemblages are changing across time. So we want to keep going, and we've done 2025 now, so there'll be one to add. I mentioned that we did beach seining at the beginning. So this is the, just give you a sense of the overlap between what is detected in the eDNA and what's detected in the beach seins in terms of fish species. We also just take the details to also note, because sometimes the eDNA sequences are only, we can only get them resolved to genus or family, but in those cases, they might still overlap with the fish in the beach seine. So this is a new visualization we're working on just to show that overlap. So it's fairly similar to what people find generally in these kinds of studies. And another note is that the fish that we don't see in the eDNA that are in the beach seine, the gray moons, those are, they're always low abundance in the beach seine. I'm kind of trying to indicate it here with these numbers. This is the proportion of each of these fish species out of the numeric proportion of those fish species within the whole catch. So they're low abundance. So we kind of know that from other studies. The eDNA is not good at detecting low abundance fish. And that's usually what we miss. Okay, another thing we can look at is sample completeness. So because we can look at the occupancy pattern of fish detected across samples, then we can use that to get a sense of how close we are to seeing all the fish. It involves some assumptions and it's borrowing ideas from Cow et al. But these are sample completeness curves for every region and every year. So in some cases, we might see that we're not close to 1. That's probably because every time we look, we see a new species. And in other cases, we're getting really close to 1 and we're sort of saturating. So that gives us a sense of how close we are as well. Another thing we can look at is those latitudinal ranges of fish that I was sort of drawing on the first frame. We can look at the blue bars here is the latitudinal range from the eDNA. So this is the whole latitudinal range here on the y-axis. And the light bars are the latitudinal range of those same fish. They're right beside the blue bar, the same fish, but from what we know from the literature. So how they're described in a lot of books and checklists from the coast. Of course, we expect to see some patterns. One thing to say right away is these fish on the right, each of these fish species, we definitely are doing better at seeing their full ranges with the eDNA than these fish on the left. So we've dug into that a little more, and I can talk about that. But I can tell you they're the fish that have better detectability within sites. So and then we can look at biogeographical patterns. So these are all the occurrence records from OBIS and GBIF for the same species we've detected in the eDNA. And this is just showing PCOA1. So it's like the ordination of, it's giving you a sense of how the community composition is changing with latitude. That's from OBIS and GBIF. So those are records, 20 or 30-year grabs of records of those fish species. And we do the same kind of graph from the eDNA. We see that we can recapitulate that biogeographic pattern. We're not really too surprised by that, but it's reassuring to see that we're seeing the biogeographic patterns that we expect. Okay, this is a part where I just don't want to get it too complicated, but it's really exciting because now we can take for all these fish species, we can pivot to ask what their thermal affinity is based on their global, because we know the fish species name, we can find out where globally it's distributed, and we can take the thermal range of that fish. We can say what's its mean temperature, what's its most extreme hot, what's its most extreme cold, and we can start to look at patterns in those thermal affinities. So this plot is showing for every year the thermal affinities, the mean temperature occupied by every fish. So every point is a fish species found at this latitude and this site. And so what you can see, I hope, is that there's this big transition in thermal affinity from warm affinity down below point conception to cold affinity just above point conception, and then they kind of stay at that cold affinity. We really have this long stretch of the same thermal affinity. And that was a surprise to me. It makes sense when you think about biogeographic breaks, but somehow I was expecting just the fish to just gradually change in their thermal affinity, but instead, no, we really have a big range. Hot, temperate fish have a long range of sort of similar affinity fish up in the, at least the Northeast Pacific. When we did ask, because we have 4 years of data, we could ask, did the mean thermal affinity of any of these communities change when there was a warmer temperature in the water? So this is sort of the kind of thing we had hoped to look for. We only found that signal, it was only 4 years, but we only found that signal in one location. And the location is kind of interesting, given the pattern of thermal affinity that I just showed you about. The location, maybe you can kind of guess where that location is. I'm going to tell you, it's right here. So just north of that major transition zone, that's the place where we're seeing the fish assemblage respond to warming in the way we expect. So either warm fish are coming in or cold affinity fish are going out. Okay, I'm not going to go through this figure too much, but just to say, we can also identify fish species that are already within our range, that they're already in the top 5% of their thermal range. And so for those fish, if you're looking at this graph, it's the ones left of these, so all these fish here, anything left of these crosses. So those are fish that we expect will move in the next heat wave. So now we can make some forecasts and see if they bear out in our subsequent years. I put this at the end because people were interested last time. We have another project where we're asking about power, how much sampling effort is required to detect change with eDNA. It's coming down the pipe soon. The main idea here is that if you expect to see a big change in your, let's say, richness, you don't need as many samples. If you want to detect a small change in richness, you need more samples. That we kind of know, but I think in the eDNA world, we're sort of grappling in the dark a little bit. What we've done is we've built through simulation and use of some of the theory, we've built our package that allows you to put in user-supplied species by sites data, and it could just be pilot data, and then you get a sense of how many samples you need, which is we call coverage, but how many actual samples you need, and how you can also put in a cost per sample for a given level of change that you want to detect at a specified power. Super happy to nerd out about that if you're interested, but Jake is going to be speaking at the Living Data Conference later in October, if anybody's going to that, and sort of revealing this work that he's working on, as well as Eden. Okay, that's all from us. Thank you so much. And a big thank you to our huge network and to our funders, especially Hakai and the Tula Foundation.
Sorry, I was taking some notes and then I couldn't find my unmute button. Thanks very much, Jen. Are there any quick questions? You just answered mine, which was going to be about the funding, but you said that right at the end. So, because I don't know if I'd missed that before. Is that kind of long-term funding, or is it kind of wait and see what happens from year to year?
Yeah, it was a 5-year commitment that ended this summer. So
Okay.
we're waiting to see what happens from year to year. Yes, fervently writing grants right now. I think this is super fundable. I just, like, as you could imagine, doing, I don't know, there must be a word from this when you're in the middle, where you're like, I don't have the funding to hire to delegate. So we're like, I'm doing a lot of the communications and we have a small team doing the data analysis. So it's hard to also take the time to write the grant. Happy to commiserate.
Okay. Well, thanks very much again. I think we'll move on to, I don't want to run out of time for the last presentation. So thanks, Jen, and we'll move on to Nastassia. So staying on the West Coast, Nastassia is from Scripps Institution of Oceanography, and we'll be talking about the West Coast OBON project.
Thanks, Sophie. I'm just going to share my screen. And yes, so as Sophie mentioned, my name is Nastassia Patin. I work at Scripps, and specifically I work for a program called CalCoffee, which is a joint NOAA Scripps ecosystem monitoring program, California Cooperative Oceanic Fisheries Investigations, and we are trying to leverage this program to bring on eDNA as an important monitoring tool. But Our project is a little bit different from the other projects. We are, we're not really a scientific project per se. We're more of a network of projects. So we sort of recognize that there are all these different institutions and programs along the West Coast of the US, including 2 folks that we just heard from in Canada. And there, a lot of these are starting to use eDNA. And so our goal is to bring everyone into the fold of OBON and make sure we are all harmonizing our methods and speaking the same language when it comes to eDNA and pushing the science forward, so that we can then bring our different data sets together and actually present these larger regional analyses of biodiversity on the West Coast. And so as part of that, I do want to recognize my co-leaders of West Coast OBON who couldn't be here today, but are crucial components of the whole network, the whole team. And so many of you probably know Dr. Susie Thoreau at the Southern California Coastal Water Research Project, Aaron Satterthwaite, who's also with Cal Coffee here, and Zach Gold, who is now with NOAA PMEL up in Seattle. And so we try to bring our own home institutions and you know, so that we can sort of lead the way in this network. Because we do have this larger goal, as I mentioned, of unifying, or rather, you know, maybe not unifying, but bringing together all these different observing systems and monitoring programs, so that we can identify our knowledge gaps and our method gaps, and then do the work to fill those gaps with intercalibration experiments or larger comparative studies. And then using the results of those studies, we can move forward so that we all adopt the best practices that we've come up with and agree upon, with the larger ultimate goal of using these best practice methods to address a whole suite of scientific and management questions for conservation, for preserving our biodiversity, and just moving forward in this age of climate uncertainty. So we do have a background, a website that I always want to point to if you're interested. We do post job opportunities and some information about current projects on there. So I do encourage everyone to take a look if you have more questions about West Coast OBON. And so as part of this talk, I wanted to just do a recap of our big activities and accomplishments from the past year, which it was kind of nice to go through these and realize how much we really have done. So we hosted 2 workshops. Both of them were down in Southern California. One was at SCORP and one was here at Scripps. The SCORP workshop was about the use of eDNA to better study and understand the effects of ocean acidification in our marine environments. And so we brought in both chemists and organismal biologists that were more had more expertise on the OA side of things. And then we also had eDNA experts, as well as management and even folks involved in doing sort of regional programmatic, you know, standard monitoring, basically, where they're seeing these effects of OA and wondering how we can better understand and mitigate them. So it was a really productive workshop. And we have a really nice final report that we submitted to the Ocean Protection Council that I'm happy to share with anyone if interested. And our second workshop was about Oxford nanopore technology. So I'm sure many people on this call are familiar with this, these ONT little benchtop sequencers. And we held this technology here at Scripps to just start to move our community forward and better understand how we might use this technology. When is it appropriate? When is it maybe not cost effective or appropriate? And so we had some representatives from ONT actually come and do a presentation both on the theory and the sort of the biochemistry and the technology behind ONT, as well as some hands-on demonstrations and discussions of different like library kits and protocols that we might want to use for different applications. So it was really, really productive. And then outside of the workshop, we also published this reference barcoding guide, which we're very proud of. And this is basically a how-to guide for anyone interested in contributing to efforts that have already been mentioned to improve our reference sequence databases. So as we all know, often our databases are missing crucial data on crucial organisms or groups of organisms. And the only way to get them in there is to actually go to a voucher specimen that is, expertly classified and preserved, do the sequencing from that tissue, and deposit the sequence data into these publicly accessible repositories. And so for folks that are, maybe less familiar with molecular methods or with database curation, this might be kind of a daunting task. And so we made this guide that is sort of a step-by-step approach for how to do that, and it's available on Zenodo. And then the last thing I'll briefly mention is that we several people within OBON have taken on leadership roles in other OBON groups or related working groups, and I'll just mention the one that I'm leading along with Matt LeMay from the Hakai Institute in Canada. Okay, so I've already talked a little bit about this workshop, but it was just a really great group of people that are thinking about ocean acidification from various aspects, whether it's the basic science of it, all the way to the translation into monitoring and conservation applications. And so it was nice to get some people that don't normally talk to each other about these different topics in the same room. We even had a local congressman stop by, which was cool, who is interested in these environmental issues, and I think was glad to see scientists and managers working together on this topic. And we came out with some key recommendations, which include developing some targeted assays for some key bioindicator taxa, which we have, had been identified from previous studies. As always, circling back to this goal of standardizing, harmonizing eDNA tools and methods so that we can bring together these large-scale monitoring data sets. And then another thing we talked a lot about was using existing data. Oftentimes there's a big push to do new sampling and go to new places, but the more we interact with our partners in this space, the more we realize there's actually a wealth of existing molecular data out there. And this has always been part of the goal of West Coast OBON, is to make sure that we all know what data exists already and who is in charge of them, and can we bring them together and apply them maybe in new and different ways to answer the questions of the moment. So we talked about that and how we might do that moving forward. And there was a little bit of future perspective ideas on using not just maybe genetic DNA data, but also whole genome data as well as transcriptome data. So really looking at gene expression, since a lot of the OA experimental work has focused on that side of things. So those were some sort of future directions that we would like to pick up again down the line. And this was our second workshop about ONT. This was held last January at SIO, and we had 18 attendees in total from several of our different West Coast OBON members. And like I said, we had some employees from ONT come and give presentations on the theory and the practice of these benchtop sequencers. So it was really a really neat opportunity to get some insider information. I don't know how many of you have used ONT technology, but it can be, I found, quite confusing. There's just a really wide range of kits and protocols, and depending on your goals in your experiment, it can be hard to figure out which one is exactly the right one. So this was a big step forward for us as we think more about it. And we came up with some sort of general assessments of some of the advantages and disadvantages of the technology. And we have, I think, now a nice little community of folks, not only at Scripps, but within West Coast OBON, that are interested in using it. And so hopefully we'll continue to work together to make the best use of the sequencing technology. And here's a little more information about our reference barcoding guide, which, as I mentioned, is available now on Zenodo. and has some really beautiful graphics and photographs, many of them taken by our own West Coast OBON members. But it was really, the development of this guide was really a process that included experts at all levels of the reference sequencing process. So we talked a lot to our colleagues at the Natural History Museum of Los Angeles and the curators of the collections here at Scripps that are very, their whole job is to ensure that these biological specimens are well cataloged, taxonomically identified, preserved appropriately, all of which are really crucial components to getting high quality DNA and ensuring that the DNA is linked properly to its organismal information. And so, so we started with them, and then, you know, moving through our experts on molecular biology methods, including DNA extractions and Sanger sequencing for full-length marker genes, as well as a big emphasis on mitogenome sequencing. I think Kristen had a really good, had some really good points about mitogenomes and how they are, you know, becoming really a critical part of building up these reference databases. And now with the sort of accessibility and low cost of mitogenome sequencing, we're really pushing for that to be a major, you know, goal of folks that are interested in improving our reference databases. So we talk a lot about mitogenome sequencing compared to marker gene sequencing, and why we think that is going to be, you know, generally the best way forward to really build these robust databases. And there is a statewide initiative here in California to build an intertidal biodiversity DNA barcode library. And so this was, funding for this was approved last year, and it's sort of in the nascent organizational stages at the state government level, but we are really hopeful that our guide will provide a framework for everyone involved in that project to really make sure that the barcoding that occurs is really high quality and as informative as possible. So the Global OBON project meeting last November, one of the big outcomes from that was these new working groups that can start to really, I think, drill down into some of the big overarching topics, some of which I've already mentioned, including things like data synthesis, bringing together different data sets, policy, technology, and then I wanted to focus on reference libraries. As you can tell, this is a big focus of West Coast OBON, and so I was excited to help lead that along with my colleague Matt LeMay at Hakai Institute. And so we had our kickoff meeting, and we, you know, OBON has this nice framework for sort of identifying how we can best accomplish our goals, called the theory of change. And so this is, I'm presenting here a very simplified version of this, but essentially you start with, well, you can start in different sort of areas of the flow, but you might start with a vision and then work backwards to say, you know, what do we need to accomplish that vision? You could also start with a current state and knowledge gaps, which is sort of how I've organized it here. And so in the case of our reference libraries, you know, we might say that, well, some issues with our current databases is that they're either too broad, like GenBank, or they can be very highly restricted. And so it's hard to try and, you know, use multiple databases to make sure you are covering all your bases. And so to address those, we need databases that are easily accessible. They're not something that is technologically, you know, where you need a computer science degree to access. They also need to be appropriate for your geographic location and range. That could mean global, or it could mean very, very regional, and both can be appropriate under different circumstances. And something else we talked about is the importance of identifying intraspecific variability and recognizing that maybe one sequence, one barcode sequence from one voucher specimen does not necessarily represent the genetic range of a target organism. And so this came from discussions of sort of our own struggles with metabarcoding and how we have reconciled those. And then finally, we have some action items that came out of that that are both scientific in nature as well as more logistical and coordinating with other groups around the world, including the Marine Technology Society in the US, OBON in Europe, and eDNA Aquaplan, and even organizations like BOLD, which is a fantastic database that has continued to push this field forward and look for new ways to improve their database and their tool. And so we're going to be discussing this more at the upcoming projects meeting, but I think we are really well positioned now to sort of tick off some of these action items and get some real improvements in the reference database space. So we at West Coast OBON, we continue to try and push these bigger projects forward, as well as focusing on smaller, more local efforts. And so while we want to continue our coordination with our other OBON projects and colleagues, including BeBOP, which you'll hear about in a second, HIBON, the Hakai Institute, FAIR eDNA, and that can include scientific studies where we're co-authoring manuscripts together. And we're also just promoting these efforts at our home institutions. So here at Scripps and Cal Coffee, I try to really spread the good word of OBON and the importance of using some of these tools that we have developed to bring our eDNA community together and sort of in line methodologically and in the scientific goals. We'll, I'm sure, continue the capacity building that we are doing in terms of holding workshops and also promoting the use of eDNA for conservation and management. And so that can mean writing grant proposals to state and regional monitoring programs that push forward the use of eDNA because it really has been proven to be so effective and efficient and accurate. And so we want to start seeing that put in use at the managerial level more. I've talked a lot about reference libraries, and then finally just strengthening our international collaborations, especially now in the United States. It's a really challenging environment for ocean science generally, I would say. And so we really value these international collaborations that exist within OBON, and we really hope to continue building new ones. And so I'll take any questions with that.
Thanks very much, Nastassia. It was great to hear about these workshops that I remember you mentioned that they were on the horizon when we had the last annual meeting. So it's great to have that update. Has there been any report from the ocean acidification workshop, or is there anything online?
Yeah, there is a report. I don't know if it's online. We wrote a report that was submitted to the Ocean Protection Council, which is a California state government group. I could probably reach out to them and ask about sharing it more broadly, or maybe sort of make a pared down, like synthesized version and share it with OBON. So I can definitely look into that.
Yeah, that would be great, because I know at the last meeting we had a presentation by a colleague on the Global Ocean Acidification Network, Observing Network, GOON. And
Yeah.
I think they were quite interested in finding out more about the outcomes of that workshop.
Yeah, that's right. Yeah, thanks for reminding me. I'll look into sharing the report or some version of it. Great,
thank you. Just to highlight, thanks, Jen, for answering the question from Mary. So that's in the chat now. So I think we'll move on to Katie Pitz from MBARI, who will be presenting a project that's been mentioned a few times in this session, BeBOP, which has formally finished as an endorsed UN Ocean Decade project, but is still, I guess, active and still very much part of the OBON family. So looking forward to hearing your updates, Katie.
Thank you, Sophie.
We can see your screen, your slide.
Hi, everyone. My name is Katie. I'm at MBARI, and the work I'm presenting today is kind of more similar to Nastassia's project in that it's bringing together a lot of people from many different institutions, all working towards trying to make how we share omic protocols better. And so our project is called Better Biomolecular Ocean Practices, or BeBOP. And we have technically ended as an endorsed project, but this year we've been making some exciting updates and working on a publication to share all the work we've done. And so still hoping to move forward kind of all the products that have been an outcome of this project. And so I'd like to thank everyone over the years who's contributed and who've also helped with this recent publication. So first, I'd like to start out, why do we need a project like this? And so first major one is that when we're talking about understanding and interpreting environmental DNA data, you really need to understand the variability in the protocols that was used to generate that data, because we know that minor differences in protocols, such as what filter size you use to filter the water, or differences in PCR protocols or primers, can have large effects on the communities that you're getting out of your eDNA data at the end. We also need to know which types of data we can join together. And so part of that has to do with how that data is generated and knowing when it's appropriate to lump two types of data sets together to make bigger conclusions about regional or global patterns. We also want to improve reproducibility of our science. So we want to have version control of methods associated with our data and detailed instructions for how someone new might try to implement our protocols. And then we also want to make our protocols themselves more findable and transparent. And so we all know, there's limited space in a publication maybe to describe the protocols you're using. And often when we come to publish data and publish protocols, we may have already moved on to a new protocol, or maybe there's been changes. And so it's really important also to be able to see what protocols are people using right now when they're collecting their data and analyzing their data. And so we thought, especially when considering omic time series, that's kind of a key consideration. And so one of the tools we decided to use for this project was to create these Markdown protocol templates. And Markdown is a computer language, so it's machine readable. But we envisioned that for each of your workflows, like a typical eDNA workflow, you'd have a different protocol for each of your steps, allowing you to track exactly how that data was generated. And this idea we worked on for this whole duration of the BeBOP project. What's kind of been new this year is that in our Markdown templates, we've started directly incorporating different metadata fields. So we pulled in not just metadata about the protocol itself that we developed as part of BeBOP, but also we've started collaborating with the FAIR eDNA project. And so we're taking their rich metadata that they're recommending for these different steps in the omics metabarcoding and other eDNA pipelines, and directly embedding it into our Markdown protocol template as what is called YAML front matter. But basically it's making a direct link in the same document between all the metadata and the values for that metadata that are about the step that protocol is then describing. And here it's showing the different sections of the protocol that are contained within our BeBOP template itself. So why is that important? Well, we're really excited about it because we see this as a workflow where you could have your protocols shared and stored in BeBOP templates alongside your FAIR eDNA, which then makes it easier if you then move to when you're sharing your data through OBIS or the INCC or across other databases. And within the FAIR eDNA project, they're working on tools to try to make this pipeline easier. And particularly within the NOAA Omics program, they're starting to implement many of these, both BeBOP and FAIR eDNA standards. So, what's better about our solution within BeBOP? It's a way to detail out the exact protocols you used. We have that version control we mentioned where we're hosting these Markdown templates on GitHub and tracking changes like you would code. Also through GitHub and Zenobo, you can long-term archival and get a DOI assignment for your protocol. And then everything is machine readable, so structured in a way that allows you to easily parse them and use them with different computational tools, and also make it easier to compare between different groups and protocols that different groups are using, making protocols more findable and accessible. So part of this is the metadata terms that we've defined within MIAP, which we call minimum information about an omics protocol. But here we've just binned protocols into different methodology categories to place them in that workflow, and then defined a set of different terms that relate to the goals of the protocol, where the protocol was designed to be implemented, different requirements of that protocol, and then terms that help us catalog the protocol. And so we envision in the future with the values from these terms, as well as values from FAIRY terms, for example, it could help you sort and find protocols more easily. So what does it look like on the actual platform? Here's a screenshot from GitHub of a protocol that was written from Luke Thompson's group at NOAA-AML. But earlier when I was saying that we directly incorporated the metadata fields, this shows up online as a scrolling bar table where you can see the term and then the value for that term within the protocol itself. And then the text of the protocol begins. When you look at the actual code of what that looks like in Markdown format, it's a list. So it's a list of terms and the values for those terms that have been provided by the author. And so this is how we'll be able to harvest those terms from the protocols when it comes time to submit your data. So different sections of BeBOP protocols are specific to the type of protocol you're describing. So here, for example, I just chose like a PCR protocol where we ask you to highlight the sequence of your primers and the PCR workflow, but it's just an opportunity to give some standardized formatting to make it easier to compare between protocols, between groups. We also ask you to fill out an equipment list to allow those who are new to your protocol to understand what kind of equipment is needed. And so altogether, kind of what this gives you is both the metadata that you need alongside kind of a detailed representation of the protocol that's all machine readable. And we hope in the future to help put it into the pipeline of the software tools that are being developed for the FAIRY eDNA project. One other, I think, nice product that's happened is as labs have started to incorporate BeBOP protocols, it's been nice that they've developed like lab-specific landing pages linking all their different BeBOP protocols in the different categories from sample collection, extraction to library prep. And we've had for a while now a GitHub page that we've used to collaborate to move this work forward. And this is where we store our protocol templates that are empty and can be forked to your own individual repositories. And also it's where we've worked on our own MIAP metadata standard. So here's just an example of where the empty templates live. And so, yeah, to I guess bring it home again, what our eventual workflow will be is that, this year we spent a lot of time thinking about how to incorporate these different metadata standards into the protocol itself. And then we see it then being a chain of these protocol documents for a particular workflow that results in your results and your data. And so we've been working with the FAIRY eDNA project to see how the software tools that are in development for the FAIRY project can link up with what we've been working on, and how that'll make it easier for scientists to then deposit their data alongside the appropriate metadata and representing their protocols in a very detailed way. So that's it for our project updates for this year for BeBOP. And I'd like to thank everyone who's put a lot of hard work into this project. Thank you very much.
Thanks, Katie. It's great to see in your presentation and the others how all the different OBON projects are working together, and that particularly BeBOP and making eDNA fair, that you're not kind of working in parallel, but actually intersecting. So that's great. Are there any questions? Jen?
Katie, how can can you give me a sense of how long, like, assuming, because we kind of have all of those things. We're at the stage exactly right now where we want to do everything that you just said. And either I'm going to do it or I'm going to have a student might like do it. How long would it take? Like, assuming we have all the information, it's just like stick it in.
I think the hardest part is having the information.
Okay.
So if you have having the information, and then I do think it is an investment in time to create a BeBOP protocol. Like when I've done it, if everything is already there, then maybe it's only half a day or like several hours, but it's basically moving the text over into the template. I think the hard part is when you don't have that information or don't know what to enter. And so that's actually prompted many of us to realize we weren't tracking appropriate information that we needed to earlier on in our protocol change. So that's been a good thing that it's made us realize, oh, okay, we need to pay attention to this because this is a fairy metadata term or because we should be saving that in a BeBOP. But yeah, it's not an insignificant amount of time to create the first one. But then I think what we found is when you then create versions of that protocol, or say like you have one PCR assay, now you're creating your second PCR assay, then that's much easier because you're just saying, here's my different primer. And maybe Nastassia can share too about, she's been going through that effort as well.
Well, I was just going to say that I think what's nice now is that there are several different groups that have a range of protocols from DNA extraction, sample collection to PCR protocols. And so it's pretty likely you can find one that is like already somewhat similar to what you need. And then that makes it much easier to then just modify an existing version. So I think the early days were like, yeah, when we all had a variety of different types of protocols at different steps of the workflow, it was harder. But I think you could probably, a motivated student, that knows how to use Markdown could do it in a day easily.
I have a question to all of you. Do the protocols somehow characterize the uncertainty in the data that you're getting?
Any volunteers to tackle that one?
Yeah, I guess my gut reaction is no, not explicitly, but I think we've all come to learn about different common primers that are being used in the area. And so I think that's something we're building up on, like saying like, when you use MyFish, you can't distinguish between rockfish species, or I don't know, I guess you're kind of like learning about uncertainty on that level of what primers may be good or bad at detecting for different on the species level. I think when you're thinking about uncertainty in the data in terms of space and time, I think Jen, you brought up, how that's an area of work right now, and that I think we're all interested in exploring and learning more about that.
Yeah, I can speak to that a lot. I love that question, Jay. Because we thought about this a lot, obviously, and there's I think there's two, maybe there's other kinds, but the two main kinds of uncertainty I'm thinking about is that annotation, or like which species is this sequence associated with, and the other is false negatives, like what am I missing? Am I sure that to get a sense of the detectability. And so with the nice thing about almost all of these programs is people do replicate. It's easier, so much easier to replicate. And so than in more traditional sampling techniques. And so with replication, I think we have a handle on sampling uncertainty. Our big problem is the annotation uncertainty. And yeah, we also got all into like, can you maintain, I don't know, like a the actual pipeline by which you do the annotation. I think those are, everyone could say what they did, but there's also there's always a million choices that could have led to something else or something else. We had a postdoc who was trying to take all the possible options and have this like distribution of possible assemblages. And it was kind of cool, but it was too much. Maybe one day we'll get into that. But he was trying to exactly like carry the uncertainty all the way to the end. But those are my thoughts on that. That's a good question.
Okay, thanks for that. Any final question in the last 3 minutes? If not, I think we'll wrap up. It's getting quite late here in Europe. So thanks again to all the speakers for those great presentations. It was really, really nice to get those updates on OBON projects. So thank you for your time, and looking forward to seeing some of you in Hobart in a couple of weeks, and in Hobart or online.